We're staring down a massive global challenge, right? How do we keep warming below that crucial 2 degrees C threshold from the Paris Agreement?
Absolutely. And it's becoming clearer and clearer we absolutely need carbon dioxide removal, CDR technologies.
Yeah, just cutting emissions isn't, well, it's not going to be enough on its own.
We simply can't just reduce and hope for the best. We have to find ways to actively pull CO2 out of the atmosphere.
Yeah.
And one approach that holds significant promise is enhanced silicate weathering.
Enhanced weathering.
Yeah.
And the potential there is big.
It's potentially huge. Estimates suggest it could mitigate maybe 2 to 4 gigatons of CO2 per year globally.
Gigatons. That's an incredible scale. And what's really compelling, I think, is that it's not some totally artificial tech. It's mimicking this powerful natural process.
Exactly. It's something the Earth has been doing for, well, geologic history to stabilize the climate.
So we're just trying to speed it up.
Pretty much. Think of it as accelerating Earth's own carbon cycle. It's fairly basic chemistry, really, but on a grand scale. When certain rocks are exposed to CO2 dissolved in water, they slowly break down.
Okay.
And this reaction releases positively charged atoms. We call them cations. Things like calcium, magnesium, sodium, potassium. And as part of that, bicarbonate is also formed. This bicarbonate dissolves in the water, and that's effectively storing the carbon.
In the water itself.
Initially, yes. And then potentially, over time, those cations can react further with the dissolved carbon to form solid minerals like carbonates.
Locking it away for good or, for a very long time.
For geological timescales, potentially. Much more secure storage.
Okay, so rocks plus CO2 plus water equals carbon storage. Simple concept. But here's the twist I find really interesting. What if we could get a little help?
From biology.
Exactly.
Yeah.
From the microscopic world. What if tiny organisms, microbes, could act as catalysts? Could they significantly speed up this naturally slow process?
And that's the core idea behind bioenhanced weathering, giving nature a nudge with biology.
Which brings us right to today's deep dive. We're going to unpack a really interesting research paper that looked at exactly this. They focused on 2 specific types of volcanic rocks, a lapilli from Gran Canaria and a commercial lava basalt.
Right, quite specific materials.
And they tested how effectively a specific mix of microbes could boost their weathering rate and, crucially, their carbon capture, all in a controlled lab setting.
Using these small flow-through columns.
So our mission today, let's walk through this paper. Understand their setup, what they found about weathering speed, how much carbon got captured, and then, what does it all mean?
Yeah, what does this tell us about using bioenhanced weathering as a real CDR strategy? Let's get into it.
All right, let's start at the beginning. The setup. How did they actually test this in the lab?
Okay, so they used what are called flow-through columns. You can sort of picture them as small cylinders, almost like big test tubes maybe,
right,
filled with the rock material. And the key is they could control the flow of water through them and then collect and analyze the water that came out the other end, the leach it.
Got it. So what went into these little rock columns? What were the ingredients?
Each column had 100 grams of one of the 2 volcanic rocks, either the Gran Canaria Lapilli or the commercial lava basalt.
Okay.
And then to some of the columns, they also added 2.5 grams of finely ground wheat straw.
Wheat straw. Why add straw?
Good question. That was basically food for the microbes. An organic carbon source specifically added to support the growth and activity of the microbial culture they plan to introduce.
Ah, makes sense. Fuel for the workers. And to see if the microbes actually did anything, they needed controls, right? Different versions of the column.
Exactly, you need comparisons. So they set up 3 main types. First, the biotic columns. These had the rock, the straw, and they were inoculated with their specific mix of microbes, the full treatment. Second, the abiotic columns. These were the controls to see the effect of just the rock and straw together without microbial life. So rock, straw, but instead of live microbes, they added sterile nutrient broth.
To account for any chemicals in the broth itself, but keep it sterile?
Precisely. And third, rock-only columns. These just had the rock. No straw, no added microbes, just the bit of slime to see how the rock weathered all by itself.
Okay, 3 conditions.
Yeah.
And this microbial team they picked.
Yeah.
Any specifics?
Yeah, it was a mixed culture. 4 types. Aureobasidium pullulans, Sulis variegatus, Bacillus subtilis, and Cupriovitis metallodurans.
Wow, quite a mix.
And it's worth noting these were all chosen partly because they're well studied and classified as risk group 1 microbes. Minimal risk in the lab.
Okay, good to know. So columns built, stuff added, microbes are sterile, liquid in place.
Yeah.
What was the day-to-day? How did the experiment run?
They flowed autoclaved distilled water through the columns regularly 5 days a week for a total of 6 weeks, like a little simulated rainfall maybe. And each week, they collected the water coming out the leachate and analyzed it for all sorts of chemical components. Then, at the very end, after the 6 weeks, they took the columns apart and analyzed the solid stuff left inside too.
Okay, setup clear. Let's dive into the results. First big finding. How fast did the rocks actually dissolve or weather?
Yeah, that's the first crucial measure, the weathering rate. It tells us how quickly that rock matrix is breaking down, releasing those mineral components we talked about.
And that breakdown, that release, is directly linked to the carbon capture, right?
Yeah.
Releasing flands, forming bicarbonate.
Exactly. They go hand in hand. And the core result here, it was, well, pretty clear and quite impactful.
What did they see?
The columns with the microbes, the biotic ones, showed significantly faster weathering rates compared to the abiotic control columns, the ones without the microbes.
Significantly faster? Like how much faster are we talking?
They measured rates that were about 2 to 3 times faster in the biotic columns compared to the abiotic ones.
Whoa, 2 to 3 times. That's not trivial. That's a really substantial boost.
It really is. For something like CDR where you need scale and speed, that kind of enhancement, well, it could be a game changer.
Definitely. Was the statistics solid on that?
Absolutely. Their statistical analysis showed that the presence of the microbial inoculation explained most of the variation they saw in weathering rates between the different columns. It was the main driver.
Okay. Do the type of rock matter for weathering speed? Lapilli versus basalt?
Yes, that also had a significant effect, though it wasn't quite as dominant as the microbes. But generally, the Lapilli columns tended to weather faster than the lava basalt columns.
Interesting. Lapilli faster than basalt.
Mm-hmm. And they also saw, kind of as expected, that the weathering rates tended to decrease a bit over the 6 weeks in all the columns.
How is that? Just using up the easy bits first?
Pretty much. The most easily weathered surfaces or mineral grains react first, and then things slow down a little. Fairly typical.
Okay. And I think I saw a note in the paper about the rates maybe being underestimated.
Yeah, that's a good point they raised. Their calculation was mostly based on alkalinity release in the leachate, that outflowing water.
Alkalinity, sort of like the water's ability to neutralize acid, linked to the bicarbonate.
Exactly. But if some of those reaction products, like the released cations or even some bicarbonate, got stuck inside the column, maybe bound to the straw or just retained in the pores,
then the amount coming out wouldn't represent the total amount weathered.
Right. The true total weathering might actually be higher than they measured just in the leachate, so potentially conservative estimates.
Fascinating. Okay, so microbes definitely speed up the rock breakdown. But did that translate into the main goal, actually capturing more carbon?
Right, that's the key question. So now we look at the inorganic carbon. You've got the dissolved stuff in the water, the DIC.
Dissolved inorganic carbon.
And potentially solid inorganic carbon, SiC, like new carbonate minerals forming in the column.
And the total is TiC.
Total inorganic carbon, TiC, the sum of both. That represents the total CO2 that's been converted and stored.
So what was the verdict on ticket? Did the microbes help there too?
Yes, the main finding mostly aligns with the weathering rates. Overall, the biotic columns, the ones with microbes, produced more inorganic carbon, both DIC and generally higher TiC, compared to their matching abiotic columns.
Okay, so speeding up weathering did seem to mean more carbon capture overall when microbes were present. But how did the two rocks compare for total carbon captured? Because you said Lapilli weathered faster.
Yes. And this is where it gets really interesting and maybe a bit counterintuitive at first.
Okay.
Despite weathering faster, the Lapilli columns didn't capture the most carbon. The lava basalt columns actually outperformed the Lapilli when it came to total inorganic carbon sequestration, the TiC.
Wait, really? So the faster weathering rock captured less carbon overall?
In terms of total inorganic carbon, yes. The order they found was basically lava basalt biotic columns sequestered the most TiC, then came the Lapilli biotic columns, which were actually pretty similar to the lava basalt abiotic columns. And then, the real surprise. The Lapilli abiotic columns actually showed a net loss of carbon, negative TT.
Hold on. The Lapilli columns without microbes lost carbon. How does that even work? Shouldn't weathering always capture carbon?
You'd think so. But it highlights a key difference in the starting materials. That carbon loss in the Lapilli abiotic columns was mainly because solid inorganic carbon, probably carbonate minerals, already present in the raw Lapilli material.
Stuff that was already there.
Exactly. That pre-existing carbonate was dissolving and leaching out. So without the microbes, the process was actually releasing stored carbon faster than it was capturing new carbon from the CO2 dissolved in the water.
Wow. Okay. But the biotic columns, the ones with microbes.
They did achieve positive carbon sequestration. So the microbes seem to not only prevent that loss seen in the abiotic control, but also manage to facilitate some new carbon capture on top of that.
Okay, so microbes help the lapilli stop losing carbon and capture a bit, but they help the lava basalt capture a lot more, even though the lapilli weathered faster.
That is weird. It really is. The lava basalt biotic columns sequestered roughly twice as much DIC and T compared to the lapilli biotic columns.
Twice as much.
Yeah. And they did find that strong positive correlation, though. More dissolved inorganic carbon, DIC, in the leachate generally tracked with higher weathering rates. So dissolving the rock was definitely linked to getting carbon into solution.
Okay, so we need to unpack this. How were the microbes doing this? Making such a big difference in both weathering speed and the amount of carbon captured, especially favoring the basalt for total capture.
Right. So the researchers looked for direct proof the microbes were actually active and doing things.
Like checking if they were even alive and growing.
Exactly. They used a technique called PLFA analysis. It measures specific fatty acids from microbial cell membranes, giving an estimate of the living microbial biomass.
Okay.
And it showed significantly higher microbial biomass in the biotic columns compared to the abiotic controls. So the microbes they added were definitely there and thriving. Inoculation explained most of that difference.
Okay, they were alive. Were they actually getting up close and personal with the rocks?
Yes. They used scanning electron microscopy, SEM, to get really high magnification images.
Cool. What did they see?
They saw extensive colonization microbes living all over the rock surfaces and the formation of biofilms. These are like sticky communities of microbes.
Right on the rock particles in the biotic columns.
Directly on them. And, crucially, these biofilms were completely absent in the abiotic controls, which also confirmed their sterilization methods worked for the controls.
Okay, so the microbes were alive, growing, and literally sticking to the rocks. How does that help break down rock faster? What are the mechanisms?
Microbes have this amazing biochemical toolkit. The researchers suggested several ways these specific microbes might be enhancing weathering, things like oxidoreduction reactions, changing the chemical state of elements.
Okay.
Producing acids, that's called acidolysis, which can directly dissolve minerals. Releasing special molecules called cytophores.
Cytophores.
Yeah, they're molecules that are really good at grabbing onto metal ions like iron and pulling them out of the mineral structure, like little chemical claws.
Love it.
Or even just physically disrupting the surface as they grow and wedge into tiny cracks, especially within those biofilms.
And you mentioned the biofilms as being in direct contact important.
Very likely. Many of these mechanisms, especially the chemical attacks like acids or those cytophores, work best right at the mineral surface. So forming a biofilm, basically gluing themselves to the rock, is probably key for efficiency.
Makes sense. And the specific microbes they chose, Bacillus, Cupriovitis, etc., was there a reason for that particular team?
Yes, it wasn't random. They were picked based on previous research showing they have abilities relevant to breaking down silicate minerals.
Like specialists.
Sort of. Like Bacillus subtilis is known for general mineral breakdown. Cupriovitis metallodurans is interesting for iron cycling and biofilm formation. Oribasidium pullulans is good at sticking to surfaces and weathering things like quartz. And Sulis variegatus is a fungus known for making organic acids and taking up metals from rocks.
So, a carefully curated team of potential rock weatherers.
Exactly. A strategic mix.
One of the things microbes breathe, right? They produce CO2 through respiration. Could that CO2 have played a role by dissolving and making carbonic acid, helping the weathering?
That's a really good point. Microbial respiration definitely adds CO2 to the local environment within the column. And yes, that dissolved CO2 forming carbonic acid would contribute to weathering, potentially lowering the pH right near the rock surface.
Did they measure that contribution specifically?
In this particular study, they didn't quantify that specific effect, say, using labeled carbon isotopes to track microbial CO2 versus atmospheric CO2. Their focus was more on the overall net effect of adding the microbial community versus not adding it, but it's almost certainly part of the enhancement mechanism.
Okay, got it. Let's circle back to that really intriguing result. Lapilli weathered faster, but lava basalt captured significantly more total carbon, TIC. How do we make sense of that?
Right, it seems odd at first glance, but the difference seems to boil down to the distinct properties of the two rocks, and crucially, how those properties interacted with the microbes, especially when it came to the form the captured carbon took.
Okay, what were the key rock differences they looked at?
Well, first, surface area. Interestingly, the ground lava basalt particles actually had a higher specific surface area than the Lapilli particles they used.
So more area for reactions on the basalt.
You'd think that would explain faster weathering, but remember, Lapilli weathered faster overall. When they calculated the weathering rate per unit of surface area, like how much alkalinity was produced per square meter of rock.
Normalizing it.
Exactly. When normalized, the Lapilli still showed faster weathering rates. So for a given amount of surface, the Lapilli material itself was inherently dissolving more quickly.
Okay, so even with less surface area overall, the Lapilli minerals were just easier to break down. What about the actual minerals inside the rocks?
That was the second key difference, mineral composition. While their overall chemistry, like silica content, was similar, the specific types and amounts of minerals differed.
How so?
The Lapilli just happened to have a higher percentage of minerals known to weather relatively quickly at lower temperatures, things like anorthite, a type of feldspar, olivine, diopside, a pyroxene.
Easy targets for weathering.
Kind of, yeah. The lava basalt also had some of these, but it had higher amounts of other minerals like nepheline, leucite, and orthoclase, which might weather a bit differently or slower under these conditions. So that mineralogy difference likely explains why Lapilli started weathering faster.
Okay, Lapilli had more fast weathering minerals. But again, faster initial breakdown didn't equal the most carbon captured in the end.
And this brings us to the third point, which seems to be the real clincher. How the microbes influence the fate of the carbon, particularly the formation of solid inorganic carbon, Si.
The carbonate minerals.
Exactly. Remember how the Lapilli abiotic polyms actually lost that pre-existing SiC?
Yeah, it was leaching out.
The microbes in the Lapilli biotic columns managed to stop that loss and capture some dissolved carbon, DIC, leading to a positive overall balance. They didn't seem to trigger much formation of new solid carbon minerals.
Okay. And the basalt.
In contrast, the lava basalt biotic columns did show significant accumulation of new SiC. They actually measured it in the solid material at the end.
So carbon was precipitating out as solid minerals in the basalt columns with microbes.
It seems so. And when they took this SiC accumulation into account, it actually boosted the calculated weathering rates for the lava basalt, because forming carbonate consumes alkalinity. This made the link between total carbon captured, TiCa, and the weathering rate much stronger and clearer for the basalt compared to the Lapilli.
I think I'm getting it. So the lava basalt might not have dissolved quite as fast initially, but maybe its specific chemistry or the ions it released, combined with the microbial activity, created better conditions for that dissolved carbon to actually lock away as solid carbonate.
That seems to be the most plausible explanation. The minerals in the basalt, maybe releasing more calcium or magnesium perhaps, interacted with the microbial processes in a way that favored precipitation of SiC.
Whereas the Lapilli dissolve fast, releasing ions. But maybe those ions or the conditions weren't quite right for forming lots of new solid carbonates with these microbes.
Exactly. So the microbes were seemingly more effective at promoting that crucial step, turning dissolved carbon into accumulated solid carbon in the lava basalt environment compared to the Lapilli environment. And that's why the basalt ended up winning on total inorganic carbon captured.
Wow. Okay, this is fascinating stuff, but it's all happening in tiny lab columns. Why should you, listening right now, care about these specific rock microbe details?
Because this kind of research is absolutely fundamental if we're serious about developing CDR methods that are safe, effective, and importantly, scalable.
This isn't just academic curiosity.
Not at all. It's direct proof of concept. It shows that bioenhanced weathering, specifically using volcanic rocks like these and a targeted microbial community in a system where water flows through. It's a viable strategy. It works, at least in the lab.
And the key takeaway seems to be about the specifics.
Critically, yes. It highlights that you can't just grab any rock and any microbe. The specific combination of the rock feedstock you choose and the microbial inoculum you use is paramount.
Because their interaction dictates everything.
It dictates the effectiveness. Both how fast the rock breaks down and how much carbon actually gets captured, and maybe even more importantly, in what form easily re-released dissolved carbon, DIC, or much more stable solid carbon, SiC.
And that point about the Gran Canaria Lapilli, the fact that it's already used locally in agriculture, that seems relevant too.
Very relevant. It suggests potential synergies. using materials that are locally abundant, perhaps already being mined or moved for other purposes like agriculture, maybe materials known to support microbial life. That could be key to making this more practical and sustainable on larger scales.
Finding those local opportunities.
Exactly, leveraging local resources and possibly integrating with existing land uses.
Okay, so to wrap up this deep dive then, this research gives us solid evidence that adding the right microbes can significantly boost how fast volcanic rocks weather.
Speeding up that natural process.
And this accelerated weathering, in turn, enhances the capture of carbon from water flowing through the system.
Turning atmospheric CO2 via dissolved forms into stored forms.
But the specific type of rock makes a huge difference. The rock that weathers fastest isn't necessarily the one that captures the most carbon overall.
Right, because the interaction between the rock's chemistry and the microbe's activity influences whether that captured carbon stays dissolved or gets locked away more permanently as solid minerals.
It's that rock-microbe partnership that determines the final outcome for both speed and storage. A really crucial piece of the puzzle for developing effective CDR.
It really is. It shows the potential, but also the complexity we need to understand.
So a final thought for you, our listener, to chew on.
Yeah, building on that complexity. This study, as we've said, used small, controlled lab columns over just 6 weeks. What are the big hurdles and also the exciting possibilities when we think about taking these findings out of the lab and into the real world?
Like, how do you scale this up?
Exactly. Could we realistically apply this in, say, agricultural fields, maybe mixing rock dust with soil microbes, or in managed forests, or maybe even in specially designed engineered reactors?
And how do you find the perfect match?
Right. How do we optimize these pairings, specific rock types with specific microbial communities for all the different environments, different climates, different soil types out there? How do we make sure we're maximizing carbon removal and ensuring that carbon stays locked away safely for the long term?
It really highlights the journey, doesn't it? From a promising lab result to actually making a difference for the climate on a global scale.
It does. It raises that big question. What's the path look like from this kind of proof of concept to a widespread, truly impactful climate solution? Lots to think about.